Good morning, students. Today we are going to start the next topic of chapter number 13, photosynthesis in higher plants. We have already read about what does we know about photosynthesis. Now we will read where does it takes place. If I ask you where does photosynthesis takes place, you would answer in the green leaf or in the chloroplast, as you have earlier read in chapter number 8. You are definitely right, but photosynthesis is not as simple as we have read yet. So photosynthesis thus takes place in the green leaves of plants, but it does so also in other green parts of the plant. Can you name some of other parts where you think photosynthesis may occur? Yes, it may be a green stem of a cactus. You would recollect from previous unit that the mesophyll cells in the leaves have a larger number of chloroplasts. Usually, the chloroplasts align themselves on the wall of the mesophyll cells such that they get the optimum quantity of incident light. When do you think the chloroplast will be aligned with their flat surface parallel to the walls? When would they be perpendicular to the incident light? You have studied the structure of chloroplast in chapter number 8. Within the chloroplast, there is a membranous system consisting of grana, the stroma lamellae, and the matrix stoma. There is a clear division of labor within the chloroplast. Here is the structure of chloroplast: outer membrane, inner membrane, because it is a double membraned organelle. Grana are attached to each other by stromal lamellae. Stroma is there, ribosomes are there, lipid droplets, starch granules. These are the things that are composed inside the chloroplast. There is a clear-cut division of labor. Within the chloroplast, the membrane system is responsible for trapping the light energy and also for the synthesis of ATP and NADPH. In stroma, enzymatic reactions synthesize sugar, which in turn forms starch. The former set of reaction, since they are directly light driven, are called light reactions, photochemical reactions, but the later are not directly light driven, but they are dependent on the products of light reactions, that is ATP and NADPH. Hence, to distinguish the later, they are called by convention dark reaction. It doesn't mean that darkness is required, but light is not required. So it is carbon reaction. However, this should not be construed to mean that they occur in darkness or that they are not light dependent. They are indirectly dependent on light, not directly. Next is how many types of pigments are involved in the photosynthesis. Looking at plants, have you ever wondered why and how there are so many shades of green in their leaves, even in the same plant also? We can look for the answer to this question by trying to separate the leaf pigment of any green plant through paper chromatography. This is the experiment, or this is the technique to separate the pigments. A chromatographic separation of the leaf pigments shows that the color that we see in the leaves is not due to a single pigment but due to 4 pigments. Chlorophyll A, that is bright or blue green in chromatogram. Chlorophyll B, that is yellow green. Xanthophyll is yellow, and carotenoids are yellow to yellow orange. Let us now see what roles these various pigments play in the process of photosynthesis. Pigments are substances that have an ability to absorb light at specific wavelength. Can you guess which is the most abundant plant pigment in the world? Yes, chlorophyll A. It is the most abundant plant pigment because it absorbs light of different wavelengths. We will see the absorption spectra in the figure 13.3a. Of course, you are familiar with the wavelength of visible spectrum of light as well as the Vibgyor. Here is the figure representing the absorption spectra of light by different pigments. We can see the chlorophyll A, carotenoids, and chlorophyll B. Chlorophyll B, it absorbs different wavelengths. Chlorophyll A different, and carotenoids different wavelength. From figure 13.3a, can you determine the wavelength at which chlorophyll A shows the maximum absorption? Does it show another absorption peak at any other wavelength too? Yes. Which one? So let us see the two peaks of the absorption peak of the chlorophyll A. Now, in the figure 13.3b showing the wavelengths at which maximum photosynthesis occurs, we observe that the wavelengths of photosynthesis, maximum photosynthesis and absorption of chlorophyll A are the same, and that is the blue and the red region. So it shows the higher rate of photosynthesis. Hence, we can conclude that chlorophyll A is the chief pigment associated with the photosynthesis. But by looking at figure 13.3c, can you say that there is a complete one-to-one overlap between absorption spectrum of chlorophyll A and the action spectrum of photosynthesis? Let's see. Here is the rate of photosynthesis. We obtain the two peaks at the same wavelengths as the two peaks were obtained by the absorption spectrum of chlorophyll A. See the next figure, which shows the black one is rate of photosynthesis and blue one is rate of absorption by chlorophyll A. We can see first peak at 450 nanometer and second peak at 670 nanometer. So these are the two wavelengths of red and blue light. So we can say that maximum photosynthesis occurs by chlorophyll A. So it is the major photosynthetic pigment. These graphs together show that the most of the photosynthesis takes place in the blue and red region of the spectrum. Some photosynthesis does take place at the other wavelengths of the visible spectrum. Let us see how this happens. Though chlorophyll is the major pigment responsible for trapping light, other thylakoid pigments like chlorophyll B, xanthophyll and carotenoid, which are called secondary or accessory pigments, also absorb light and transfer the energy to chlorophyll A. Indeed, they not only enable a wider range of wavelength of incoming light to be utilized. True, क्योंकि अगर क्लोरोफिल A होता तो सिर्फ दो ही वेवलेंथ को अब्जॉर्ब करता, लेकिन क्लोरोफिल B, कैरोटिनॉइड्स और जेंथोफिल्स तीनों मिलकर और भी विजिबल स्पेक्ट्रम की दूसरी लाइट्स को अब्जॉर्ब करते हैं, जिससे टोटल इनपुट ऑफ लाइट एनर्जी बढ़ जाता है. वह अब्जॉर्ब करके क्लोरोफिल A को ट्रांसफ़र कर देते हैं. So they also protect chlorophyll A from photooxidation. Yes, वो overshadow कर देते हैं chlorophyll A को. What is light reaction? Light reaction or the photochemical phase include light absorption, water splitting, oxygen release and formation of high energy chemical intermediates ATP and NADPH. These are the four steps mainly occurring in the light reaction or photochemical phase of photosynthesis. Several protein complexes are involved in the process. The pigments are organized into two discrete photochemical light harvesting complexes LHC within the photosystem PS1 and PS2. So there are two photosystems PS1 and PS2 in which all the pigments are organized. These are named in the sequence of their discovery and not in the sequence in which they function. Keep in mind, जैसे जैसे डिस्कवर हुए उनके नाम रखे गए, ना कि उनके काम के अकॉर्डिंग उनका नाम है. The LHC are made up of hundreds of pigment molecules bound to protein. Each photosystem has all the pigments except one molecule of chlorophyll A, forming a light harvesting system also called antennae. These pigments help to make photosynthesis more efficient by absorbing different wavelengths of light. The single chlorophyll A molecule forms the reaction center. The reaction center is different in both photosystem. In PS1, chlorophyll A has absorption peak 700 nanometer. Hence, it is termed as P700. While in PS2, it has absorption maxima at 680 nanometer and hence called P680. So PS1 is P700 and PS2 is P680. The electron transport. The next. In photosystem 2, the reaction center chlorophyll A absorbs 680 nanometer wavelength of red light, causing electrons to become excited and jump into the orbit farther from the atomic nucleus. These electrons are picked up by the electron acceptor, which passes them to an electron transport chain. Here is the light harvesting complex. We can see all the pigment molecules in green colored disc photons incident on the pigment molecules. The electrons get excited and jumps from them and are accepted by the electron acceptor, primary acceptor. इस प्राइमरी एक्सेप्टर के बाद इलेक्ट्रॉन्स जो हैं वो इलेक्ट्रॉन ट्रांसपोर्ट सिस्टम को कैरी फॉरवर्ड कर दिए जाएंगे. This movement of electron is downhill in terms of an oxidation reduction or redox potential scale. The electrons are not used up as they pass through the electron transport chain but are passed on to the pigments of photosystem I. Simultaneously, electrons in reaction center of PS1 are also excited when they receive red light of wavelength 700 nanometer and then are transferred to another acceptor molecule that has a greater redox potential. These electrons then are moved downhill again, this time to a molecule of energy-rich NADP. The addition of these electrons reduces NADP to NADPH plus H plus. This whole scheme of transfer of electrons starting from PS2 uphill to the acceptor and down the electron transport chain to PS1. Excitation of electrons, transfer to another acceptor and finally downhill to NADP, reducing it to NADPH, is called Z scheme due to its characteristic shape. First uphill, then out, downhill and then uphill. So it forms a Z scheme. This shape is formed when all the carriers are placed in the sequence on a redox potential scale. Here is Z scheme shown in the figure. We can see light is incident on two light harvesting complex. First PS2, second PS1. PS2 से इलेक्ट्रॉन्स एक्साइट होकर अपहिल जाएंगे, फिर डाउनहिल जाकर इलेक्ट्रॉन ट्रांसपोर्ट सिस्टम में आएंगे और डाउनहिल जाएंगे, सेकेंड फोटो सिस्टम में आएंगे, फोटो सिस्टम 1 में, और फिर वापस एक्साइट होकर इलेक्ट्रॉन एक्सेप्टर से जाकर NADPH बनेंगे. Splitting of water. You would then ask, how does PS2 supply electron continuously? The electrons that were moved from PS2 must be replaced. Very true. जो भी PS2 से बार बार इलेक्ट्रॉन्स एक्साइट होकर निकल रहे हैं वो वापस PS2 में आने चाहिए. दिस इज अचीव्ड बाय इलेक्ट्रॉन्स अवेलेबल ड्यू टू स्प्लिटिंग ऑफ वाटर. और इसीलिए हमें फोटोसिंथेसिस में वाटर के स्प्लिट होने की ज़रूरत पड़ती है. स्प्लिटिंग ऑफ वाटर इज एसोसिएटेड विद PS2. वाटर इज split into two protons, nascent oxygen and electrons. This creates oxygen, one of the net product of photosynthesis. The electrons needed to replace those removed from photosystem 1 are provided by photosystem 2. Here is the reaction representing splitting of water. Twice of the water molecules gives 4 protons plus oxygen is evolved and 4 electrons which are taken up by photosystem 2. We need to emphasize that the water splitting complex is associated with PS2, which itself is physically located on the inner side of the membrane of the thylakoid. Then, where are the protons and oxygen formed likely to be released in the lumen or on the outer side of the membrane? This we have to study कि जो ऑक्सीजन और प्रोटॉन रिलीज हो रहे हैं वो कहाँ जाएंगे, ल्यूमेन में या मेम्ब्रेन के बाहर.